All right, guys, let's talk about Heredity Standard 1. This is going to build quite a bit on stuff we've already done in class. So we already did the web quest where we learned about DNA in general, and we did the timeline activity, which we'll expand on. This is the whole standard. If you look at it, there's sort of three parts. Gregor Mendel and the laws of inheritance, the structure and function of DNA, which was determined by many scientists, and the fact that DNA it contains genes. So we're going to look at sort of all three pieces today. So we already know a little bit about DNA. Again, we did that web quest. And we also know it was a team effort to actually discover the structure and the function of DNA. So remember, science is not like an individual thing. Scientists don't work on an island alone and like make all these discoveries. It really comes from building off scientists' work that other people have done. So scientists are constantly reading about what other scientists do in order to make better predictions about what could happen. And we've already talked about the scientists a little bit when we did our timeline activity. So I'm just going to kind of review those for you, but they should already be nice and neat in your notes. So we started with Griffith, right? He was a doctor. His patients were very sick from pneumonia, and he wanted to know why. So he was studying pneumonia. He wasn't even trying to figure out anything about DNA. And he saw that there were two strands of pneumonia, right? There was a deadly strand, and there was a strand that was not deadly. He knew that heating up bacteria killed it, right? So he heats up the deadly kind that's dead as a doornail. He combines it with the kind that's not deadly. He leaves it for a little bit of time, and then all of a sudden he has live deadly bacteria in his sample. And so he's like, what in the world? Where did this deadly bacteria come from? The only thing that was alive was the one that's not deadly. And he realized that the heated up dead strain somehow gave something to that non-deadly strand that let them change into the deadly form of the bacteria. And he called this the transforming factor, right? Because it allowed the bacteria to transform into a different kind of bacteria. He didn't know what that transforming factor was. He didn't know which part of the cell it was, right? But that was his research. Next came Avery. So Avery was like, all right, this is interesting, but what is the transforming factor, right? So he wanted to know what it was. So he took that same bacteria and he started to destroy parts of it to see if transformation could still happen, right? So he destroyed the protein, transformation still happens. He destroys RNA, transformation still happens, right? He destroys lipids and carbohydrates, other stuff, transformation still happens. He destroys DNA, no transformation. So that tells Avery that in bacteria, the transforming factor, genetic information, is DNA. Right? Now, Avery's experiment only covered bacteria. We didn't know if genetic information was the same in all things. So we just knew after Avery that in bacteria, the genetic information is DNA. Next came Hershey and Chase. They wanted to know if this is true in just bacteria or if it's true in other living things as well. So they studied viruses called bacteriophages. And what they did was they tagged parts of the virus with radioactive substances. So a virus is kind of just made up of a protein shell with an inside core of DNA. So there's two main parts to a virus, protein and DNA. And what they did is in one batch, they made the protein radioactive. In the other batch, they made the DNA radioactive. Right? And so when a virus comes down to infect, what it does is it injects in something to the cell, and that thing is the instructions on how to build new viruses, right? And so they watched after the injection what was radioactive outside the cell or inside. And they found when they made the protein radioactive, the outside of the cell was radioactive. So protein wasn't going in, protein didn't have the information. When they made DNA radioactive, the inside of the cell was radioactive. So that told them that viruses insert DNA into a cell, and that contains instructions on how to build a virus. And so we now know that DNA is the genetic material in viruses as well as in bacteria. And viruses and bacteria are super different, so we were then able to conclude that DNA is the genetic material for all living things. All cells use DNA for genetic material. Next came Chargaff. So Chargaff wanted to know more about the makeup of DNA. And so what he did was he studied a bunch of different species and he looked at the nitrogenous bases present. And what he would do was measure the amounts. And he found that the amount of A always equals the amount of T, and the amount of C always equals the amount of G. So in every single species he looked at, whether it was bacteria or a plant or an animal, whatever it was, the amount of A always equaled T, C always equals G. And that's called Chargaff's rule. Hopefully you realize why Chargaff's rule is true, right? Think to the base pairing rules. What does A hook with? T. What does C hook with? G, right? Chargaff's rule. Next came Franklin. So Rosalind Franklin used X-ray crystallography, which is like taking a picture of the DNA molecule. It's very complicated, lots of radiation, which caused her to die early. But she was looking at DNA, and she was able to get this really beautiful picture of DNA. And what she realized, was that DNA is double-stranded. So we didn't know that before. So she realized there's two strands to it, and they're hooked together by nitrogenous bases. So inside that double strand, the nitrogenous bases sit in the middle. So these were really helpful hints on what the entire structure of DNA looked like. Next came Watson and Crick, right? So they saw Rosalind Franklin's research, and this is kind of a sore subject because they didn't give her credit. They just pretended like they hadn't seen her work, when really it was her work that helped them figure out the structure of DNA. So a little bit sketchy, and it wasn't until much, much later we realized Rosalind Franklin was part of this. But Watson and Crick had been trying to build a 3D model. So they knew the parts of DNA, right? So if you think about DNA, there's 3 parts to the nucleotide. There's that sugar, there's nitrogenous bases, and there's phosphates. I'm going to kind of draw this out real quick, right? So there's 3 parts to DNA. Again, you have a phosphate group, a sugar, and a nitrogenous base. And so they realized that the way DNA hooks together, and again, they used Rosalind Franklin's work a little bit, right, was these nitrogenous bases were in the middle, all the way down. The sugar and the phosphate group were along the outside. So they're the first ones to determine the structure of DNA. So now we know the full-on structure, right? So DNA is a double helix. Again, that means there's 2 strands and they're twisted together. If you look in the picture, right, the outside, the backbone of DNA is made of sugar and phosphate. The inside, the rungs of the ladder, are the nitrogenous bases. And again, we now know they bond C to G, A to T. And these nitrogenous bases bond with hydrogen bonds. And this is important because hydrogen bonds are pretty weak. And we need to be able to open DNA up, right? Think about replication. In order to copy DNA, you have to open it up. So those hydrogen bonds in the middle are important that they're weak, right? And here's what it looks like again, in sort of a space-filling molecule here. So again, you have a backbone of sugar and phosphates, and you have rungs of nitrogenous bases. We now know the function of DNA, right? So DNA is responsible for maintaining all the information needed for the cell to function. So it includes the instructions on building every protein the cell will ever need. So if the cell needs more smooth ER, it's going to pull from the instruction book, from the DNA, to know how to build it. If the cell needs to build more polymerase, it's going to pull from the DNA. And it also gets passed down to the next generation. So that's DNA. All right, now let's talk about our good friend Gregor Mendel. So Gregor Mendel was an Austrian monk, and in his sort of spare time, he loved to study science. So back then, monks often were also scientists, which is kind of cool. And Gregor Mendel had a whole garden full of pea plants. And that is where he really started studying genetics without even knowing genetics was a thing. So Mendel was awesome. He worked before any of our other scientists we've talked about. He learned about all of this without even knowing that this thing called DNA exists. Basically, he's a rock star, all right? And what Mendel realized is peas were a great place to start studying genetics. They were quick to breed, so you could study them very easily, quick turnaround for generations. They had a lot of offspring, which is important because genetics is all about percentages. And the higher number of offspring, the better your numbers look. And they're self-pollinating, which means they can fertilize themselves. So what he did was he waited until he had several true breeding plants. This means whenever they self-pollinated, they had the same trait as their parent. And a trait is just a specific characteristic. So for example, if they had a round seed shape, if they were true breeding, they always had offspring with round seed shape. So here's the traits he studied. Again, lots of different things he was looking at, and they all had these 2 different traits to them. Okay, so he was studying a lot. Again, rock star. All right, so he started with his P generation, and his P generation, which stands for the parent generation, those were those true breeding plants. And he bred them to make hybrids. So he crossed parents to get different traits. So for example, he would take a plant that always made round seeds and a plant that always made wrinkle seeds, and he would cross them. So he would pollinate them to see their offspring. So that was his P generation, right? He crossed them to get the F1 generation. So the F1 generation is the babies, right, of the P generation, and those are those hybrids. And then the F2 generation are the offspring of the F1 generation. So if you kind of imagine this as people for a second, right, the P generation is like your grandparents, F1 generation are your parents, F2 is you, right? So it's 3 generations that he was looking at. All right, so what he saw, again, he took those true breeding plants, right? Like a purple flower always makes purple offspring, white flower always makes white offspring. He crossed them, and in the F1 generation, the hybrids, he found that 1 trait seemed to disappear. So 1 trait got passed down, in this case the purple one, 1 trait, the white one, disappeared. So his F1 generation, 100% of them showed 1 of the 2 traits. The other trait, gone. All right, he then took that F1 generation, this helped him realize that the traits of an offspring were based on their parents. So he called this thing a gene, something that gets passed from parent to offspring, and it gives them their traits, okay? So he didn't know what the gene was, right? We didn't even know DNA exists, but he knew there was something that was passed from parent to offspring, which was Mendel's first law of inheritance, which is the principle of dominance. Some alleles are dominant and some alleles are recessive. So an allele is a form of a gene. All right, so some forms of genes are dominant, some are recessive. If we think back to the example we were looking at, purple flowers were dominant, white flowers were recessive, right? So when he crossed purple and white flowers, 100% of the offspring showed the dominant trait. So that one is dominant. So dominant means you just need 1 allele to show the dominant trait. Okay, so if it's big H, little h, right? Let's do hitchhiker's thumb, big H, little h. The dominant trait, you just need 1 big H. So big H, and big H, little h will both have a straight thumb. The recessive trait, having a hitchhiker's thumb, right? You need 2 alleles to show that, little h, little h. So dominant allele, you only need 1. If the plant doesn't have a dominant allele, if it has 2 recessive, it'll show the recessive trait. So if mom and dad here are true breeding, always green, always yellow, the result is all green. That tells us green is dominant, yellow is recessive. So the F1 generation, again, 100% of them showed the dominant trait. So here's our P generation, true breeding, and here's our F1 generation. 100% of them showed the dominant trait. All right, so then what Mendel did was he took that F1 generation and he allowed them to self-pollinate. All right, so 2 purple parents, basically 2 axial parents, which means flowers off to the side, yellow, round, etc., to get the next generation. So think about it for a second. What happened to that recessive trait? Is it gone? Is it hidden? Is it never going to show up again? Is it going to reappear? What do you think happens? All right, so Mendel was shocked that when he got his F2 generation, 75% were dominant, but 25% showed the recessive trait. That trait showed up again. Remember, it wasn't since way back in the P generation he saw it. In the F1 generation, they were all dominant, and now 75% are dominant, but 25% are recessive. That's crazy, right? For you guys, you might already have kind of figured out what's happening because you might have knowledge of Punnett squares, but for Mendel, he doesn't even know what DNA is, right? He's like, what? Where did this recessive trait come from? He has no idea. He's shocked by it and thinks it's really cool. All right, so Mendel figured out that there's something called segregation happening. So when a parent makes their sex cells, so makes gametes, which are sex cells, right? In humans, they're called egg and sperm. He realized those alleles get separated, right? So if this is our original generation, or excuse me, this is the F1 generation, they have a dominant allele from, let's say, mom, recessive allele from dad, right? And they're true breeding, or they're self-pollinating, so they're giving up two alleles. They can either give the dominant or the recessive, right? Both parents can give either the dominant or the recessive, and those can combine randomly. And so what you see is that 75% end up with a dominant allele, so the dominant trait, 25% show recessive. So segregation, again, is this idea of these alleles separating from each other. And that's Mendel's second law of inheritance, the law of segregation. So again, during gamete formation, which is this process called meiosis, we'll learn more about it later, alleles separate from each other, so only one of them gets passed down. And here's another picture to kind of show you that. Again, if this is mom's egg, right, she has two different number 5 chromosomes, right? She could pass down either one. Same thing with dad here. This also led to his third law of inheritance, which is the law of independent assortment. So this tells us during meiosis, those genes segregate independently of each other. In other words, just because you're dominant for one trait doesn't mean you're going to be dominant for another, right? So when those genes line up to separate, it kind of looks like mitosis, if you want to picture that for a second. They line up independently. It's not like all the dominants on one side, recessive on the other. They line up randomly. And this will kind of show you. So this is what meiosis looks like. So you have your original cell, S phase has already happened, they're replicated, right? And then they're going to separate randomly. So at the very end, you have all kinds of different possible haploids, different possible gametes or sex cells. All right, so let's go through some modern genetic terms real quick. All right, so homozygous means two individuals, or an individual that has two of the same alleles. So like big R, little r, right? So they can be homozygous recessive, have two recessive alleles, or homozygous dominant, two dominant alleles. So capital is dominant, lowercase is recessive. So if they're homozygous recessive, they'll show the recessive trait. Homozygous dominant will show the dominant trait. The opposite of that is heterozygous, two different alleles, right? They're also known as hybrids, right? And that means they're going to have a big R, little r, but they'll show the dominant trait. Because remember, as long as you have one dominant allele, you show the dominant trait. So big R, little r, big R will look the same. Genotype versus phenotype is another word we need to be familiar with. Genotype is the genetic makeup. So that is, are you homozygous dominant, heterozygous, or homozygous recessive? Phenotype is physical characteristics. So do you have a red flower color or a white flower color? So remember, if you know that a flower is white, you know its genetic makeup is little r. If you see that a flower is red, it could be big R, or big R, little r. All right, so that's what we're going to cover today. Make sure you go back to the assignment to see what else you need to do, and have a wonderful rest of your day. We're going to go over all of this tomorrow in class, so don't worry if it's still a little confusing. Thanks, guys.